So greetings all and welcome to the Science of Childhood Cancer lecture series hosted by the St. Jude Comprehensive Cancer Center. I'm Madan Babu and I'll be hosting our speaker today. So I'm a member and the Endowed Chair of Biological Data Science in the Department of Structural Biology. I'm also the Director of the Center of Excellence for Data-Driven Discovery in the Structural Biology Department here at St. Jude. While this is our final lecture of the season, planning for the fall has already begun and we are excited to announce the next session is scheduled to begin August 11th. With another exceptional lineup of speakers. So please don't forget to check our website for updates and stay tuned. So it is my great pleasure today to welcome our speaker, Dr. Babis Kalodimus, who I have known for over a decade through his seminal work on protein dynamics. But before I formally introduce our speaker, just a note that you can submit questions at any time during the talk using the chat function and sending questions to all panelists. I'd also like to let you know that if there are additional questions, Dr. Kalodimus has kindly offered to stay on for some time after the top of the hour if anyone would like to stay for further discussion. And now I'm delighted to introduce Dr. Babis Kalodimus. So Babis received his PhD in 1999 from the Institut Curie in Paris and began exploring NMR as a postdoctoral researcher in Utrecht. Babis was recruited to St. Jude in 2017 as chair of world-class structural biology department here at St. Jude. And Babis recruited me recently to join his department a year ago, and it has been an absolute pleasure to be part of the department under his leadership. Now, Babis's lab scientifically aims to understand the structure and dynamics of the complex protein machineries that underlie key biological processes in health and disease. His group uses powerful NMR methodologies and has made fundamental discoveries that have changed textbook paradigms on protein dynamics by revealing hidden conformational states of kinases that are now being leveraged for drug development. As you will see in his lecture titled The Conformational Landscape of Protein Kinases in Physiology and Disease, his work opens new horizons in cancer research by illustrating the importance of rare conformational states of biological molecules, notably how the lowly populated conformational states that his group detected for the first time are actually being leveraged for the design of selective inhibitors. His scientific contributions have been acknowledged with several awards, including the 2017 Raymond and Beverly Sackler International Prize in Physical Sciences and his election as a 2020 Fellow of the Biophysical Society. So without further ado, I would like to invite Dr. Kalodimus to deliver his lecture. Thank you, Babis.
Thank you very much, Madan, for the introduction. And of course, thank you for having me here. It's a great pleasure to be giving this seminar. And let me get started here by sharing my screen. Thank you again for having me, and thank you for this opportunity to speak in this what apparently appears to be like a very very interesting series of seminars and lectures. So I will be in fact speaking today about two related but slightly different topics. The majority of my time I'll spend in fact on recent work we've done on the anaplastic kinase, receptor kinase, lymphoma kinase, and I will then also spend like few minutes towards the end of my seminar to give you in fact some idea about like a larger kind of effort we are undertaking here to understand all protein kinases, the entire human kinome, by looking into what Madan referred before as to these rare conformational states that we do really now know very well that the underlying regulatory disease mechanisms, drug resistance mechanisms, and so forth. Okay, so let me get started here by first sharing with you some of our recent data we have on a very interesting receptor kinase that has remained until recently, at least in terms of the structure and how this kinase is activated, unknown. We have now solved this puzzle. We have addressed some questions, and then as of course always happens, once you dig deep, then new questions also emerge that we hope to be able to address them in the near future as well. Now, ALK, or anaplastic lymphoma kinase, is, as I said before, a receptor kinase, and there are about 60 of them, 58 to be precise, that they all share some very common features, right? So they all are membrane proteins. They have a single pass helical part here, and then all of them have a kinase domain in the cytoplasm, and then in the extracellular region, which is, as you can see here, different kinases. They have like different domain organization. They have different motifs, and this is because they bind to different ligands. So the whole idea of RTKs, or receptor tyrosine kinases, is to signal from outside the cell to inside the cell, and they do so by interacting, by binding to ligands, and then through a variety of mechanisms, then the kinase domain changes its structure, becomes phosphorylated, and as such, it does really then contribute to signaling inside the cell. Okay, so with as the basic underlying principle here with all receptor kinases, this is kind of like an oversimplifying picture, because you know now there are divergent mechanisms when it comes to how RTKs are activated. But the principle is that we have receptor tyrosine kinases in the absence of a ligand. They typically exist in a monomeric state, although it does not really apply to all RTKs. For example, insulin receptor kinase is an exception here. But in this monomeric state, the kinase domain adopts an inactive state. And as such, of course, you know, like it's not capable of phosphorylating. And then when you have a ligand, the ligand binds to the extracellular region of an RTK. They bring the two kinases, the two monomers, if you will, close nearby as a dimer or larger oligomers. And then when this happens, the two kinases, the kinase domains in the cytoplasm, they interact with each other. They phosphorylate initially their activation loop. They now convert into an active state, and as a result, also typically also additional phosphorylation events are really happening on the C-tail or the transmembrane or this juxtamembrane region here of RTKs. These phosphorylation sites, then they create docking sites. So for other signaling molecules that they come and dock and interact, and of course that will then create a relay of interacting molecules, and as such you have, this is how you get across the message, and then that can signal outside or even inside the nucleus within a cell. So this is the basic underlying principle, as I said before, although we know the divergent mechanisms. Now, when it comes to ALK, it has, of course, an interesting story, and then it also has a story, like to St. Jude, which is very interesting because ALK was discovered here at St. Jude, like almost 30 years ago. Then and it was discovered initially as a fusion with NPM. And then, as I'll show you actually in a minute, since then, many more fusion proteins with ALK have been discovered that are involved in all kinds of different disease. Now, physiologically, not much is known about ALK. We know that if you knock them out, not really much is happening. We also know that the expression is severely reduced after the early months or years of humans. Nevertheless, we also know that it can really signal through some of the very well-known and characterized signaling pathways that you can see here. Some of them are, you can see this RAS MAP kinase pathway or the PI3K. These are different docking molecules that they come and then they interact, scaffolding molecules with ALK. But other than that, much of the physiology, I think, remains still to be discovered. There's been some recent also data that reported ALK as being, for example, involved in obesity as a thinness gene and so forth. Much more is known, though, unfortunately, in a way, about ALK, simply because it seems to be a hotspot for reasons that are not really well understood, but these are a hotspot for the creation of these ALK fusion proteins. And then we have many of those, and in the majority of the cases, these fusion proteins, they cleave the extracellular domain also the transmembrane helix, so then becomes cytosolic. And then these two, the fusions here create a dimer. So they bring these two kinases together, and as such, now ALK becomes constitutively active because its activity is not anymore regulated by ligand binding or unbinding or, like off and off, as you would have under physiological conditions. Now, this is the variety. We have over 20 well-characterized fusion proteins we have, oncogenes with ALK, and you can see some of the examples here, as well as, like the different cancers that they cause or they are involved in. In addition to these ALK fusions, also there are many point mutations. We know also, for example, that overexpression of ALK is related in some cases with disease, although it's not very clear whether, of course, they are driver or they are somehow involved in the disease progression or not. It's not really as clear-cut as in the case of the fusions that we have with ALK. Nevertheless, there are many of these mutations that, again, most of them are found in the kinase domain. And, like neuroblastoma is one of the very, like notorious kind of like cancer that ALK, the fusion of ALK is involved in. Okay, so this is an introduction here to ALK. And like moving forward, as I said, one of the big mysteries until recently is how ALK looks like, what is the structure, especially of the extracellular region, that nothing was known about it, but also how then it becomes activated under physiological conditions. So this is the domain architecture of ALK, and then I also introduce here LTK, which is a leukocyte tyrosine kinase, because they are in the same family. This is a transmembrane helix here, of course, a kinase domain, but when you look at the extracellular region, they are very, very similar. So this part here of ALK is very, very similar. It's actually over 70% similar in terms of sequence to LTK. So then we do really expect that the structure of these two kinases in this region would look very similar. And it's also possible that some of the activation mechanisms or how they, for example, they bind and interact with the ligand, they might be, very similar as well. ALK also has additional domains here on the N terminus. It's a much, as you can see here, it's a much larger protein than LTK. It's about, as you can see here, 1,600 residues. LTK is about half of that in size. Now, ALK also, together with LTK, until recently, were part of what we call these orphan receptors that a ligand had not been identified. And that changed, in fact, just a few years ago when in the lab of Joseph Schlesinger at Yale, as well as actually, in the lab of Ruth Palmer's as well, they identified two ligands, two physiological ligands, that they interact with both ALK and LTK, and they activate them. So, for example, you can see here, like you add what we call the augmenter alpha or beta, and then both of, like you can see, they can activate both ALK and LTK as well with like, to a different extent and degrees here. And an interesting finding here, I think, is also the fact that augmenter alpha, and there are also like different names for these ligands. It can be a little bit confusion, right? For example, I guess the accepted nomenclature is ALK-IL2 and ALK-IL1. I will refer to that as augmenter alpha and augmenter beta for the rest of our talk here. But alpha is dimeric. They are both very small proteins, as you can see in a minute. It's like about 10 kilodalton or so. But then the augmenter beta is monomeric. So the fact that you may have either a dimeric ligand or a monomeric ligand, and they can both very efficiently activate a receptor kinase, that is rather unusual in the field of RTKs. So that, provided, in fact, some additional questions here. But of course, it's more intriguing in terms of what kind of mechanism would really allow, either a dimeric or a monomeric ligand to bind, dimerize, that's at least the idea, and the ALK in this case, and activate it. Okay, so we then undertook this work here. In fact, Andrei Resetniak, who has been the leader of this project in my lab, was before in Joseph Schlesinger's lab when he made this discovery about the binding of the augmenters to ALK. So then when he joined my lab a few years ago, then he wanted to basically go after that and then try to get and understand structurally and mechanistically how ALK binds to these ligands and how it gets activated. Now, in order to do that, we used what we call an integrative structural biology approach. And we combined cryo-EM, we combined NMR, as well as crystallography. This is really state-of-the-art kind of approach in structural biology. And I would like to say this is a triumph of structural biology as well, because we were able to tackle what actually really has been a very, very challenging project. And then we know that unless we had access to all of these resources, like this project would not really have been possible. And it has really been possible because here in structural biology, we have an amazing lineup of tools and state-of-the-art resources. You can see here, for example, the Titan cryos here we have, and one of the most powerful NMR magnets in the world, the 1.1 gigahertz that we have here, like I said too. Now, I will spare you the structural approach, exactly how we did things. It's probably kind of like a little bit more esoteric, although it's very gratifying for structural biologists and biophysicists, I guess, to follow these approaches. So I'll just get directly to the main menu, which is how do really this, the extracellular region of ALK looks like. Okay, so we started then, and then knowing that this is the part of the kinase here that interacts with the ligand. So we refer to this one as the augmentor binding region, right, which is the ligand binding region. This is about, as you can see here, something a bit like about 350 residues or so long. And this is what is required for very high affinity for the ligand. The ligand binds to this, even the isolated ABR domain here, ECR domain, or fragment rather, with nanomolar affinity. So by using them initially, by combining NMR spectroscopy and crystallography, we were able to get this structure now of this fragment here. That's the ligand binding fragment. And then we did this one, as I said, by combining NMR and crystallography first in the absence of the ligand, right? So we want to understand, first of all, how the architecture, what is the structural details of this extracellular domain. We expected to see something unusual based on sequence. We knew that ALK had a structure, had a domain organization and a sequence that was very different than any other RTK. So then we would expect to find something novel here. And then indeed, we did find something really novel in terms of how the domains are arranged, but also, for example, how some of these domains look like. This is here in the green part is what we call this glycine-rich domain. It's a very unusual architecture. As you can see, this is what we call this glycine polyglycine 2 helices. This forms a very rigid here part of the of this ECR domain here, together with a TNF-like domain here, and then down here like a smaller domain that we call a EGF-like domain as well. Now, there are probably like details that many of you probably don't really care as much, but just like in case that some structural biologists here or some people would like to know what are the contacts that we do see here and then how they look like. This is a very rigid overall part, especially this part here. This is more flexible, and then we know that EGF likes to move, like relative to the rest of here of the scaffold, but we have some disulfide bridges that they help to stabilize the structures and then give them, especially the smaller domains, a well-folded structure here. There are disulfide bonds here as well here that it keeps things like more and more rigid. So there are contacts here that you can see. There are hydrophobic contacts. In some cases also there are some hydrophobic contacts as well. But you can see the overall arrangement of these domains, how they would like basically to exist, and the structure they adopt in the absence of the ligand. And as I said before, it was surprising the fold we got here into this glycine-rich part. There are very, very few proteins known with this kind of fold. And even for this couple of examples we have, typically you only have like a very small number of this glycine-rich, polyglycine two helices. In this case, we have, as you can see, about 14 or so. The way they pack, all of these red dots here, dotted lines here, they denote hydrogen bonds, and this is the hydrogen bonds you have across these strands or the helices, and they help rigidify to a very great extent this domain. It's still uncertain what is the role of this. I mean, why, for example, you would need to have such a rigid structure here in a fold, but I guess that's something remains to be addressed, hopefully in the near future. Okay, now going into the ligand part, as I said, only very recently the ligand was discovered, the physiological ligand for ALK. We know it's a small protein. The structure was unknown, so in this case we used NMR spectroscopy to determine the structure of the ligand. Again, of course, like in the absence of the kinase. You see here the domain organization is a small protein. Altogether, it's about 125 residues. Once the signal peptide is cleaved, it has what we call this augmentor domain, and this is absolutely required for interaction with ALK. And then there is also a variable region that has a sequence that is just differs widely. In fact, within augmentor alphas, but as well also between the augmentor alpha and beta as well. Now, in the case of, as you remember, augmentor alpha, I told you that this is a dimer, and in fact, it's a constitutive dimer because there is a cysteine here in this variable region that gets, oxidized and forms a disulfide bridge, and such, then this augmentor alpha, it is when it is expressed outside the cell, it is a constitutive dimer. This is how the structure looks like. It's a very, very simple fold, as you can see here, with a couple of helices, as well as you need to have these disulfide bridges here because it's a very small domain, of course, and you would like to have this its structure as intact and as stable as possible. So then these two disulfide bridges here, they do confer a lot of rigidity to this structure. And this is again some ideas, some of the side chains and distributions, and the many hydrophobic interactions you have within the interior of the protein that helps stabilize the ligand. Okay, so we got now the kinase in the free state, the structure of it. We also got the ligand in the free state as well, the structure of it. And now the big question, of course, is that how they come together, how do they really interact? And then the next big question is that now how the ligand, of course, activates the RTK, in this case, the kinase. So this is, I'll give you a schematic and I'll show you also like a couple of animations to try, just to get across as clearly as possible what is basically the conformational changes that are elicited by ligand, and then how we can get now a dimeric form of ALK. All right, there are massive conformational changes really happening here, so it might be a little bit trickier to convey this one accurately, but I'll try, to do this to make this one as clear as possible. Now, this is the structure here. Again, just to make sure you're all on the same page. This is the fragment that we are studying, right? We've got the structure of this, which is here, and now we have the augmentor alpha, for which also we have the structure, and then we form a complex, right? So then when augmentor binds, the extracellular region of ALK undergoes a massive, very pronounced conformational changes that moves from this conformation to this conformation. And it is basically like two coupled rotations and movements as well, where you do have about different kind of axes, right? So you have a rotation around the z-axis, as you can see here, but also like you have a very rotation, in fact, around this axis here that brings, as you can see, makes ALK much more compact than in the absence of the ligand, right? So in the absence of the ligand, it prefers to exist in this kind of like linear, elongated, rod-like structure, but then when augmentor binds, it basically makes it much more compact. It bends, and then brings these two domains much closer to each other. And you can see some of the interactions here as well. We show some of the side chains that help you probably better appreciate. There are again a lot of hydrophobic interactions, and EGF, as you can see here, this yellow part basically closes down on augmentor alpha. So then it extends by a significant amount the surface that is buried, and then as well as the interacting surface. And then although augmentor is like a very small molecule to start with, nevertheless, the affinity, as I said before, it is very high. It's about 30 to 40 nanomolar in this case. Okay, so we've got that, and then we are determining now by cryo-EM, because this becomes now a large molecule and large complex when it becomes dimeric, right? You know that we've used a lot of biophysical techniques that we know now when you add the augmentor alpha, and then when augmentor alpha binds to ALK, then we have a large complex that forms with a stoichiometry of 2 to 2. So then you have basically one part that you see here, and then you have one molecule of another ALK that they come together bound to augmentor to form now what it is, a heterotetramer, or what is an ALK dimer, you know, like in this case. But we have 2 ALK molecules and then 2 augmentor molecules as well. And this is how the structure of the dimeric extracellular region of ALK looks like. I am displaying here one of the monomers as a cartoon. So you can see the glycine region here, and the TNF-like, and EGF, as well as the augmentor alpha. And then the second dimer, or the second molecule that had the second complex with ALK and augmentor, it is here displayed as a surface, as a solvent-accessible surface, just to distinguish, you know, like the two protomers in this case. And this is just a rotation by 90 degrees or so, where you can hopefully, you know, like appreciate how then these two proteins come together to form a dimer. This is here the part that is colored red. This is the interface that is formed at the dimeric interface that basically mediates dimer formation and makes it this one, you know, like a stable complex. Now, this is a cryo-EM structure, you know, like I am, you know, like in the interest of time, I'm just, you know, like I'm not including many of the details. But that has not really been an easy structure to determine because of flexibility and because of many other, you know, like issues as well. I just want to highlight that augmentor alpha in this case is a dimer, right? It's just that this variable region, the one that mediates dimerization, is very flexible, and as such is not resolved in our cryo-EM map, but nevertheless, it is really present there. And then we know, you know, like that forms a dimer, and this is, you know, what drives, this is the main driving force for bringing these two ALK molecules together. You can also see here in this animation, probably a little bit clearer, how these two molecules come together to form this dimer. It is mediated both by augmentor to the other augmentor on one complex, on one molecule to the other molecule, but of course there's a very extended, as you can see, surface also between the ALK, the two ALK molecules as well. So then you have both ligand to the other protomer, but also like protomer to protomer contacts and interactions that stabilize the dimer. And we have, of course, as we typically do in structural biology, once we have the structure, then we also we run several assays, both biochemical, biophysical, as well as cell-based assays, you know, like to interrogate the structure and make certain that the structure that we have does really reflect the structure that we have in the physiological structure. And indeed, you know, like when we introduce mutations that compromise the dimeric interface, then ALK cannot really be activated anymore. So if we compromise the ligand receptor interface, ligand doesn't bind, and as such, of course, you know, you have no activation. But also, if you compromise here by mutations, the dimeric interface, so then now you don't really have a stable dimer anymore, then as well, in this case, ALK cannot really be activated. So we have a very well confirmed that indeed this is a structure that corresponds to the physiological structure of ALK. Okay, so now if I would like just then to kind of like summarize what we've seen in this case is that when we have the ligand, when we have this, in this case, in the absence of the ligand, we are looking at ALK, right? And this is the extracellular region here. We expect this one to be very flexible and dynamic relative to the membrane, right? Because this one here, there's like a long linear, about 15 residues or so. We know that the linear is unstructured, and as such, we really expect to have like a very high freedom, in this case, of flexibility. So then this, the ESR can really move relative to the membrane, I guess, with pretty much unrestricted degrees of freedom. Nevertheless, when augmentor binds, as I showed you before, then it elicits these massive conformational changes that you can see here. And then together with the second molecule of ALK that they come, they have now the force ALK to basically lie flat on the membrane. It adopts a conformation that is parallel to the surface of the membrane. Now, this is a very unusual architecture, never seen before, as far as you know, in RTKs. And that, like already, I think, highlights how the different and divergent mechanisms that RTKs can use in this case to oligomerize, dimerize in this case, and become active. Now, when you look at this arrangement here, right, and then again, of course, like all the work we've done, we haven't done this one with the membrane or the transmembrane helices. This would be extremely challenging. This is, what we are currently working on, and we hope that we'll be able at some point to get the whole structure in the presence of the membrane as well as the transmembrane helix as well. But when you look at this arrangement here, and then because we know that the linker is relatively short here, and then we know that this needs to sit very close on the membrane, right? So when you look at that, and you see that this augmentor here, also ligand, is expected to interact with the membrane. And that's also something very unusual because ligands that activate RTKs typically bind to domains that they sit far apart, far away from the membrane as well. So that was very intriguing. And as such, like we basically ask ourselves, is it really possible that the ligand also directly interacts with the membrane? And so if someone takes a closer look here on the augmentor, this is the augmentor part as we expect to sit on the membrane, or how they will really, the whole overall disposition with respect to the membrane. As you can see, there is a number of positively charged residues that they come very close where the membrane would really be. And in fact, if you look across the both alpha and beta augmentors, we do really see that these positively charged residues are highly conserved, right? And of course, as you know, like the surface here of the membrane is negative. If you have positively charged, then they're negatively charged. And if you have positive charges on the part of the ligand, that there's a very favorable electrostatic interaction. In this case, attraction, like of course, it would get attractive forces that would help even more stabilize the dimer, as well as also the overall orientation that we have of ALK compared to, relative to the surface. So in this case, what we did then is that we mutated these residues, we reversed their charges, right? So then we made this basically lysines to aspartates, and this is what we refer to here as reverse charges. And as you can see now, with the wild type case, you'll get like a very nice activation of ALK. But in the case where you reverse, we have now where we reverse these charges, although these charges do not affect ligand binding, so augmentors still can bind very tightly to ALK, but nevertheless it fails to activate ALK. This is in cell-based assays, and then we interpret this one as a very strong evidence that of interaction between augmentor and the membrane, which, as I said before, like it's kind of like a novel feature as well as a mechanism that we have in RTKs that further highlight the divergence of the mechanisms we have in receptor tyrosine kinases. So just let me summarize basically on what we've seen here by using this animation, which hopefully will help clarify and then also drive this home, like message that how ALK, how augmentor in this case, we use ALK-AL, like in this case, just to confuse you even more, but this is a ligand, this is the augmentor alpha that, like we've come to know and love, and this is ALK here, and you can see, given the fact that there's very good evidence that the ligand itself interacts with the membrane, this is how we envision the whole thing really happening, where augmentor gets close to ALK, now ALK undergoes this massive conformational change and poison primes it for now a dimeric interaction with another molecule. And then when this happens, right, the two monomers here, now of course they move like very close to each other, they form a strong dimer. And then when this happens, then you have activation of the ALK in this case that is based on the that is induced, of course, by ligand binding. Okay, so there are many other important details that we have included in our paper, and then if you are interested, that was published, like last December, like a few months ago. So there's a lot of details there if you want to, but let me just summarize what we think are some of the important findings we have here. We do have a novel domain architecture that not seen before in RTKs, and I did explain to you, for example, in this case, that the glycine region, the glycine-rich region is one of that has not really been seen before. In fact, it's very also novel among any other proteins, for that matter, in terms of the number of these polyglycine 2 helices we have. As I also explained, when the ligand binds, in this case augmentor, there is a very pronounced rearrangement of the extracellular region of ALK. As I explained this one before. Okay, here it is again, and how that happens. It really lies flat on the membrane. And then this horizontal overall disposition, it is a novel architecture in RTKs, which I find personally, in fact, very interesting and intriguing as well. And this is also what seems to be explaining how you can have either a dimeric or a monomeric ligand, augmentor alpha or beta, that they can bind to ALK, dimerize ALK, and activate ALK very efficiently, right? Because in this case, where you have now the ligand that can be on the surface of the membrane, right, the overall search process, it is being reduced from a 3-dimensional diffusion process to a 2-dimensional constrained process we have of limited diffusion, of course, on the surface, and that accelerates, but also, increases very, very significantly the affinity that these ligands can have for ALK. And then all the ligand in this case really wants to achieve is that when it binds, you need to induce the conformation to ALK that is dimer competent, right? So then when the ligand binds, then you have this massive conformational change. And now this basically says, I'm really ready to accept another receptor that has also ligand bound, and then let's try to form a dimer together for this to work. Now, in the last few minutes I have here left, I would like to go back and take a step back here, and then looking at the big picture, of course, and then when it comes to kinases, even, that's the case also with ALK, even with receptor tyrosine kinases that have these very beautiful extracellular regions, and as such, we like, of course, to understand activations and everything that is really happening there. But the important domain is the kinase domain. And even when it comes to ALK, of course, don't forget that all the fusions we have, okay, they delete the extracellular region. In this case, we are left with the kinase domain. The kinase domain becomes a driver. We have many mutations that really occur, like in this case, in the kinase domain. We know that many of them, for example, activate ALK, and then yet this remains a mystery. And this is not only the case for ALK. This is also the case for, I would say, all or the vast majority, if not all, of the kinases we have out there. And there's a reason for that. And then I would like just to take a couple of minutes here, highlight the work we are doing, okay, and then how we expect, together with many other kinases, but also with ALK, how we would like then to investigate the kinase domain itself, and then try to understand how mutations in ALK domain, the ALK kinase domain, how they activate the kinase, the drug-resistant mechanisms we have, the disease, for example, we have the oncogenic mutants, and so forth. So we have a very big initiative here, structural, this is a blue sky initiative on protein kinases, seeing the invisible. This is a massive undertaking, thanks to the generosity, of course, of our institution, that they really believe in state-of-the-art science, and then pushing the frontiers in many regards. And definitely this is what we are trying to do in this case with the protein kinases. It is about seeing the invisible in protein kinases, and that really goes back to the initial title that I have in my talk about the conformational landscape of kinases, both in physiology but in disease as well. As you can see, this is, as I said before, a massive project of about $50 million or so. So what is the idea of this blue sky project we have here is to basically leverage ultra high field NMR to detect and structurally characterize, and of course understand all these functional mechanisms, okay, of what we call these alternate conformational states or rare conformational states or invisible conformational states in protein kinases. We have now very strong evidence that such states that are otherwise extremely hard to detect because they are present only for a very limited time, they are fleeting structures, they do underpin regulatory mechanisms, drug-resistant mechanisms, as well as, for example, in this case, like oncogenic mutations and so forth. But also, we believe they provide new opportunities for therapeutic intervention. So, why we would like then for someone to study and then look into these conformational states? Well, this work, I have to say, it's really, like builds on a very recent work from our lab where we used Abl kinase to try then there to discover and understand the mechanisms that underlie the function of Abl kinase as a kinase, but also, for example, many of the oncogenic mutants, as well as also drug-resistant mechanisms, for example, in this case, Gleevec as one of the most, like well-known drugs for Abl. So why you would like to understand this conformational landscape? Well, we know now very well that for many proteins, and that's definitely the case for kinases, for all kinases, that they do exist in multiple conformational states. And then these different conformational states may have distinct function. And the functions can be, for example, binding to different ligands or having like different catalytic activity, enzymatic activity, or kinase activity in this case, or really may, may signal in a very different way altogether. But these different and alternate conformational states, or this multitude of conformational states, where someone would describe as a conformational landscape, is extremely hard for someone to really detect, and much more challenging for someone to structurally characterize, because as I said before, the systems are very dynamic, and as such, traditional kind of approaches are really very, very hard to apply, in this case. So this is what we would like to do now for all the human kinome. And as you know, there are over 500 of them, right? And that's this massive undertaking that we are taking here, because as you know, kinases are extremely important. They are involved in all kinds of like different signaling and biological processes, but also as it happens, like they also mutated very frequently, either through point mutations or deletions or translocations and fusion proteins, and they cause cancer. They also know that they are the target of many approved kinase inhibitors, and there are many, of course, other in clinical trials. But if you look at, like it's only a very small number of these kinases that have been therapeutically explored so far. There are all kinds of like reasons for that. And then I think one of the major issues as well when it comes to targeting kinases is also poor selectivity, because of the fact that the kinase domain in the active state really adopts a conformation that is extremely similar to from kinase to kinase, right? Because there has been, of course, evolutionary pressure here to make certain that kinases signal and then bind ATP, they hydrolyze ATP, then they bind to the substrate and they transfer the phosphoryl group also to the substrate. And for all of this really to happen, you need to have a perfectly aligned structural element within the kinase domain. And then for this reason, evolutionary pressure, of course, has selected a very specific conformation overall structure of a kinase for this real to happen. Now, any deviation you have from this kind of ideal structure here, either it's in this P loop where ATP binds, or in this DFG motif here, where the aspartate coordinates magnesium and ATP as well, or the activation loop where the substrate binds and docks to, then any deviation from that would be an inactive state. And this is, what we would like to investigate. And then to do this one, we rely heavily on NMR spectroscopy, because as we showed very recently in our Abl paper, that can really be used now to look into and then determine this conformational landscape in atomic detail, right? I think this is the really major kind of like advantage of this technique, that not only you can detect populations that may be as low as 1%, right? For example, only 1% of your molecules can adopt this conformation, and then yet, like we are sensitive enough by using NMR spectroscopy to be able to detect them, but also be able to determine the structures of this. And let me just again, within a minute or so, here say what we've done with Abl kinase, because this is that forms, like the whole process and the procedure, but also we adopt this one now for ALK and many of the other kinases as well to understand, like their mechanisms. For example, in ABLE, we determine 3 of these states that they interconvert with each other at the same time. One, it is fully active, and it is the major conformation. An inactive state 1, what we call, an inactive state 2. These all interconvert with each other. In fact, as it happens, we also discovered the conformation. It's only about 5% or so present, but that's a conformation. What we call here, this inactive state 2, is the conformation that Gleevec selectively binds to. So that, like allowed us basically to better understand how Gleevec binds, but also what is the nature and the underlying mechanisms for many of the drug resistance mechanisms. And then, of course, we can follow how the structure then changes between and among these conformational states. Just give you an idea for how, for example, how ABLE converts from this active state to an inactive state. And you can see how all this rearrangement of the structural elements take place, that they convert the kinase from active to inactive, right? And then there's all this big question here, of course, how many of these distinct inactive states are present in different kinases, and this is something that we would like, of course, to find out by studying all this big number of kinases. But just to give you an example, with Gleevec and ABLE, we were able finally, like to understand how some of these Gleevec-resistant mutations. For example, like you can see here, this is the ABLE structure, and this is where Gleevec binds, and then some of the well-known drug-resistant mutations that many patients develop when they are treated with Gleevec, some of them are really very far away. For example, this histidine here to a proline, which is so far away from Gleevec, right? This is not really easy to understand by someone just looking at the static structure, how this is really happening. But by looking this one by NMR, and then in fact, by looking into the conformation here that we have, this is just a simple experiment that we run. Well, not as simple, but definitely, extremely powerful, that detects the different conformational states. I don't want to go into details here, but like trust me when I say that by looking into these dips here, we can identify the different conformational states. And in this case, for example, in the wild type, we see that this conformation that Gleevec prefers to bind to, it is really present, right? But then this mutant, what it does, it doesn't really affect directly binding of Gleevec, right? Because it's allosteric, it's very far away from the binding site. But what it does, it destabilizes, right? And as you can see here, the population goes from 6% to less than 1%. It destabilizes the conformational state that Gleevec selectively binds to. And as such, of course, it depletes it, removes it from solution, and that makes it, like, of course, like renders the affinity of Gleevec for this mutant much lower, and as such, of course, that gives rise then to drug resistance. And then, and finally, I just want to say that by using this approach here, we are able to interrogate many of these mutations, for example, or the different structural elements we have in kinases. We definitely look forward to apply these methodologies to understand, in the case of ALK, how all these various mutations that we know, for example, people have seen in neuroblastoma or other cancers, right? How do they really activate the kinase? The structure of ALK in the absence of inhibitors remains, like it's not really known, but also, like under different conditions. So that's something that we can do hopefully very nicely once we get into this one by NMR. These are just different, very important motifs and structural elements where many of these mutations do really occur, and many of those, like convert kinases to an oncogenic mutant. Okay, so this is the key findings we have from this kind of approach we want, which is just to have been identified this multitude of conformational states or the conformational landscape, like as I said before, and then the importance of that is not only to identify and structurally characterize, but then also to leverage this information and to understand, okay, how like these different regulatory mechanisms, for example, are in play here, as well as this inhibitory mechanism. But also, I do really believe, okay, that by discovering these additional inactive states that may be really unique to individual kinases, then the chances that someone can design selective inhibitors, of course, are much higher. Okay, and then with that, let me then finish here and then acknowledge the true heroes of this work here, of the ALK work. As I said before, Andre, that has been the project of his life so far, I would like to say, although he's now to new adventures and then new kind of like very interesting things as well. Paolo was also instrumental in all of the NMR studies, and Alexander with cryo-EM, very, very challenging features. Munya helped tremendously. Darcy in the X-ray center, Amanda in the PTC center, and Norman Luo has been really amazing with his graphics. It's a great talent in graphics and animation as well. And that has been a very nice collaboration we had with Josef Schlesingers at Yale, together with his people there. So with that, I would like to conclude here. I would like again to thank you for the opportunity, and I'll be more than happy to answer any questions you may have.
Great. Thank you so much for such an exciting talk, Babis. So we have a number of questions. So I'll perhaps start with the first one from Ben Willander. So can you speculate on the role of the more distal domains of ALK upon ligand binding and sequestration and bending on the plasma membrane particularly? And are these domains important either for ligand binding or positively or negatively regulating the activity of ALK in the bound and the unbound forms?
Yeah, this is the good question. And that's something we've been discussing with Andre as well. That's kind of like the next step we'll have to take to include the entire extracellular region here in case of ALK. Of course, with LTK, there's none there. But the exact role of these domains are not really known. Of course, we know that there are a lot of glycosylation sites there. There's also heparin also can really bind, and that can further, for example, stabilize the dimer, and then also can assist in the further activation of ALK as well. But other than that, structurally, we understand some of the domains based on homology, how the structure would look like of the individual domains, but the overall disposition of that, and then whether there are interactions, for example, between the augmentor binding domain and the other domains there, that remains unknown. Someone will have to take this holistic approach and then try to structurally characterize the whole region there.
Thank you. So the next question is from Michael Schwabe. So Michael's asking, have you or others tried to use MD simulations as an additional approach to see whether one could capture some of these alternative conformational landscapes, or whether one could even parameterize MD simulations to be able to discover now that there is experimental evidence for these alternative states?
This is a good question. Yeah, there are people who have been trying to do this. There are actually several people. One of the problem that Benoit Roux, for example, is someone in Chicago has been trying for quite some time, and in fact, we are collaborating now on some of these kinases. We're trying to combine MD and NMR as well. One of the major problems generally with kinase domains from a structural point of view is that these, as I explained, these are very flexible, and we have which presents major issues when someone would try to get the structure of that. For example, the main technique behind really getting all the structure, we have over 6,000 structures of kinases now in PDB, right? And nevertheless, I would say probably like about 99% of them, or probably even more, are bound to inhibitors. And there's a good reason behind that. It's just because, when you have something which is really so dynamic, then you cannot really crystallize them by crystallography, and as such, you have to use real inhibitors to stabilize them. So that has prevented us from understanding, all these different conformational states and how they're really interconvert with each other, because when you have an inhibitor, that bias, what exactly conformation you see and you crystallize, of course, like in the crystal environment. MD can, I think, hopefully, leverage with NMR and then put this one together, because with NMR we can experimentally visualize them, and then we can see, for example, to what extent MD now can be used to predict some of those, or by training, maybe like their algorithms and whatever they really use. This is also something that, of course, have discussed for the future, given the power of AlphaFold, right? That's another shortcoming when it comes here to kinases, because the database that AlphaFold, of course, has used to train its structure prediction in kinases is heavily biased by all the structures we have that are bound to inhibitors. And as such, many, like if you ask what are inherently the structures of the kinases in the absence of inhibitors, then of course, like you don't really have that. And so hopefully through this blue sky proposal, as we're getting more of these structures in the absence, unbiased, and multitude of structures, then we can train, like these algorithms to, at some point, hopefully be able to predict all these alternate conformational states without someone, like having to do the entire experimental work that we have on that.
Great, thank you. And there's some really very interesting questions from Martin Roussel, Gerard Groswell, and Sarah Sherman. So they're related questions, so I'm going to try and synthesize it. So do you know what kind of cells express the ALK ligand and the ALK receptor? And does the ligand produced from donor cell bind to the membrane of the recipient cell before it binds to ALK?
So the what you know is that the ALK is really part of the nervous system, right? I mean, also, like, for example, it's been expressed in hypothalamus and brain as well. There's very little knowledge because once that you have the ALK, but then also until recently, and still remains unknown. For example, how about the ligand? Okay, where the ligand expresses, and there was like this very recent work, I think published in PNAS last month from Yossi Schlesinger's lab. Okay, where they found that augmentor alpha seems to to be together close with ALK in the hypothalamus, like in the brain as well. But if you knock out ALK, I believe this is maybe after a few months. We know that initially, of course, you get a lot of expression, but then when you look at the different tissues, after I think a year or so in the life of a human, then you see severely very, very little expression of ALK, like in these different tissues. And that's why I think has been very hard to understand what is the exact physiological relevance, like here and role of ALK that you really may have. There was a Cell paper published last year where they mentioned that this is, for example, related in obesity. And it's a thinnest kind of like molecule, which I think has been also amplified by the work of Yosh as well. But not really much is known. Otherwise, I think much more is known. And there's a good reason that ALK actually was discovered as a fusion kinase, right? So no one really knew that it existed until they really found out first, and they discovered the fusion protein, and that led, of course, to all kinds of like different then discoveries that we really have. Much more are known about the fusions rather than the like what is the function of the whole ALK in this case. Yeah.
Great. So we are at the top of the hour. So I'd like to thank Dr. Kalorimus for a great talk. At this time, I'd like to invite all who are interested to stay on for a continued discussion in the extended Q&A, and thank you all for coming. But before we jump back into questions, just a reminder to mark your calendars. The next session of the series will begin on August 11th and will run through November 17th. So the next question is from Juan Manuel, and his question is, you commented that about 7% of the kinome has been therapeutically explored. Do you think that the study of more treatments could help reduce the cost of these drugs?
So sorry, Madan, I missed you when you said 7% is explored, but then?
Do you think that study of more treatments could help reduce the cost of these drugs? Meaning, like, even for the same kinases, can you reuse these drugs for different diseases by targeting these conformational states?
I mean, it is well known that some of the drugs, for example, are very promiscuous, right? For example, dasatinib, that was initially designed to bind and inhibit ABL, it can be used also, and actually is really being used to inhibit other kinases as well. So there is this more kind of like promiscuous drugs there that in some cases, they work really well because they also like inhibit some other kinase you may really want to inhibit. I think it really depends on the disease and what exactly you are trying to do. But, of course, there's a great value if someone can develop very selective inhibitors, both to interrogate the biology, right? For example, if we can selectively go into the cells and then inhibit a very, the kinase that you really have in mind without inhibiting other ones, then you can learn more about, the role of these kinases, of course, in the biology, but also when it comes, like to treatment, if you have, I don't really need to lecture people in this case, but if you can very selectively inhibit, like one certain kinases, then of course, you don't have side effects, right? And then the benefits can be tremendously. I have to say also like something about ALK here is that which is very interesting because, apparently there are many, or not apparently, I mean, there are many ALK inhibitors as well that's been used, like in the clinic, like now. And these are typically well tolerated. And the reason that they're well tolerated is just because, like ALK doesn't really appear to be, required for at least, like in adults and such, like this is, I guess, a good way of treating. Although the problem, again, with ALK, as it is with many kinases, is that patients do develop drug resistance, right, to these ones, which is in many cases some of them are very obvious to understand, but also in the case of Gleevec, some of them are allosteric and as such, less obvious to understand what is the base for that.
So there's a question from Gerard, which I didn't capture in my original formulation. So his question is, can the ligand bind to the membrane without being bound to ALK? Because you were presenting this point about this 2D diffusion rather than 3D diffusion.
Right.
Are they sticky enough to bind to the membrane on their own?
Right. I mean, there's no direct evidence for that, but we do have, I would say, indirect evidence that this probably really happens. And then it is a molecule that when you try to express, purify, stabilize, it's very sticky. It has this, has this tendency, for example, like to stick to hydrophobic parts. That, tells us maybe we have, for example, to use detergents to stabilize this complex. Even, like in our NMR studies, although it looked like nice and beautiful, then Andre had to go to great lengths to make sure that, this is stable by adding detergents and so forth. So this is our speculation. Our speculation is that, yes, the molecules do really have a tendency to stick to the membrane, okay, even in the absence of like ALK in this case. Although this is, like we don't really have a very direct evidence for that, which will be very hard to get, in fact, anyway. But that's our speculation, yes.
Great. And there's a related question from Bessie and said to you, thanks for a great talk and a very inspiring work. So his question had to do with EGFR glycosylation and perhaps even considering lipid composition. So are there specific lipids on the membrane that may somehow be associated with neuronal cells that make some of these ligands more potent in certain environments? And can glycosylation of the ectodomain orient the conformation and perhaps even facilitate more stronger interactions when the ligand's present or not?
Yeah,
and is anything known about those lines?
Yeah, no, nothing. These are all great questions, but really nothing is known about that. It's of course really very hard for someone to study these things. First of all, you have to express them and purify, and then have the exact composition of glycosylation, which is extremely challenging by itself to be able to do this one, of course, in vitro. These are all excellent questions, but really nothing is known about that.
Yeah, all of these are opening up these questions, much more to be investigated. So there's one more question on NMR spectroscopy data. So I think this is a question from Ashish, who's asking, are you aware of any machine learning methods or people who use machine learning methods to complement the spectroscopy data? Are there open data sets that are available to train these ML models to be able to capture any signal from the spectroscopy data for presence of alternative conformations?
Well, I mean, is the question related to NMR spectroscopy itself, or whether machine learning's?
I think it's probably going to be about other data sets, enough data sets that are available out there to be able to train methods. For instance, with AlphaFold, we have the entire PDB, so that we can train a structure prediction method. So I think this person's question is, are there NMR data sets out there which can be used to train to discover or predict these?
Okay, to the extent I understand the question, right? Because I may be totally off here, but if there is a question, for example, to what extent machine learning, or this algorithm, like AI, can really help in predicting how, like the NMR spectra would look like, for example, and especially if you have alternate conformational states or so forth. No, I mean, the answer here is definitely no. And still with NMR, we are very far from even being able, for example, for someone to look at the structure and then from the structure predict what the chemical shifts, how these peaks and where the peaks, are really going to be in the spectrum, right? It's so complicated. And then the environment plays such a role that still, although we can make some sort of predictions, this is really nowhere as helpful, nearly actually as helpful for us to be able to use them to maybe, assign, for example, like some of these peaks and the spectra, which is very laborious now, and it's really been done in many cases manually, assisted by software. But other than that, like it really requires human intervention to a very great extent. We are not there, not nearly there. So AlphaFold has made, if there is like an alpha in MR or something like that, and then they would like this, that would be, I think, I guess, really great and amazing.
Interesting. Yeah. So we have a question from Chuck Shear, who signed in with Martine, and it was Chuck who also asked this question about what cells produce ALK ligand. I think his question is ALK a secreted ligand, meaning like, is there a paracrine way in which signaling happens, or is this an autocrine mechanism where cells expressing ALK also co-express the membrane-bound ligand? So is this intercellular communication, or is it happening in the same cell to somehow maintain a sustained signaling pattern?
The great question. But again, like my understanding, as I was trying to learn more and more about that, is that very little actually is really known, and still not very. For example, we know that augmentor beta, for example, that can activate ALK and LTK. There are some, I guess, discrepancies here in their view whether beta it is also like a physiological ligand for ALK or only LTK, because it seems to be activating much more, weakly ALK, but actually much more potently LTK. The most recent paper from Yoshi, I think, is the hardest kind of like evidence that we have when the co-expression in the hypothalamus in brain where both ALK and augmentor alpha, they seem really to be co-expressing. Other than that, personally, I'm not really aware of any other hard evidence about where exactly is located, where, and the timing, or is that really together, they are co-expressed together on what's really happening. I mean, given the fact that ALK really is not essential, and then after like few months in the in the human life, they become almost undetectable. They are very, very low expression levels. I think this is what probably just provides a big obstacle for us to understand exactly what the physiological role of that is, and also together with the ligand as well.
Great. So I'll perhaps ask the last question, Babis. I think it's probably a little bit tangential to what you presented, but nevertheless an interesting one. So you highlighted that the transmembrane helix, in the monomeric form and the dimeric form might somehow allow these helices to form a dimer. And there are so many other receptor tyrosine kinases. Has anything been done about the nature of the transmembrane helix and how exactly these transmembrane helices may even stabilize in the ligand-bound form? So that was one question. And then the second question had to do with the ligand itself. And I think you highlighted there was a variable region along with the augmented domain itself. Is the variable region important in any form, or is this just a neutrally evolving but relevant for secretion and other kinds of like homeostatic functions rather than the signaling function itself?
Yeah. Great. Good question. So the first one is, I think it's a question for the future, right? One is
yes, exactly.
Kind of like outstanding questions we have in the RTK field, and clearly, I would say the dream here is to get the structure of an intact receptor itself, right? That has proved so far impossible. And the reason for we use cryo-EM, people will have to use cryo-EM because of the size and because of the complexity, but the flexibility, it's what makes this one actually almost impossible. We can see people have done this one. Insulin receptor, for example, is one case. Like EGFR, where you look at the different domains as well, but you can see the extracellular domains here, you can see the extracellular regions, but then, for example, you miss the kinase domains. You don't really see them. They are very, very flexible. Similar problem is also with the transmembranes as well. There are being studies with just the helices themselves, but nothing else, right, where you can see them. So that has been, like around for some time. People have used, for example, in this case, an MR to be able to see the orientation of these helices, how these two helices basically pack. If we can really get to the point, and that's why we believe with Andre, we'll discuss that maybe ALK is a good example here, and maybe it's a good model system because of the constraints, because of the fact that ALK and the extracellular region, it seems to be lying flat on the membrane, and as such, like It is more constrained than in other receptors where basically they are just very wobbly, right? And then being able to capture that, it will be much, much harder. With ALK, we might have some luck here and be able to, by cryo-EM, for example, see how the extracellular region together with the transmembrane helices, okay, for example, how everything really is organized and it's packed together, right? Just because, in terms of freedom, right, they are really much actual weapon. So this is something remains to be seen, of course, and whether we can do this one in the future. And then your second question was
on the variable domain.
Oh yeah, the variable domain, right? Yeah. That's actually another interesting question as well, because again, is the difference here we have between alpha and beta, the two augmenters, that one is a dimer, the other one is monomer, right? We know that also there's been work, Dre has done work when he was with Yossi's lab, that if you use just the augmenter domain, the one that binds tightly to the ligand, that by itself can also activate the protein and ALK, so then you don't necessarily need, like the dimer in this case, which that can be explained based on our data and the fact that the ligand, for example, may interact with the membrane. But then why do you really need it, right? And then the dimer is there. But that, like raises all kind of different questions. What is really physiological? Also, is there different signaling? For example, if you have a monomeric protein binding, does it signal the same way as, for example, when a dimer is bound, right? I mean, we know now with the RTKs, it's not like an on and off thing, right? It's like much more nuanced. There are many degrees of different, ways that can really signal. We know in some cases, for example, they can the same kinase can recruit different scaffolding proteins, which probably has to do with the different level of phosphorylation. But how is that possible, right? Just having different ligands on the extracellular domain, how that's really happening. That's not really known. That's not really well understood. There's, I think, a lot of like good science that needs to be done for us to advance our understanding.
Great. Fantastic questions and excellent discussion. So we still have a large number of members here. There's one more question which has just come in from Martin again, Martin and perhaps Chuck. So were effects of ALK knockout studied during embryonic fetal development or only in surviving postnatal mice? So I think from Chuck.
I think there are in mice very early where the effect was not any catastrophic effect. There were like some phenotypes there, but not really any lethal phenotype. Even if you have like a double knockout, like as far as as I remember on that. Yeah.
Great. So thank you all for hanging around for this really wonderful discussion on this topic. Thank you again, Babis, for your talk and for answering all these questions. And look forward to seeing you all in the next session in August. See you then. Bye-bye.
Thank you, Madan.